we just get such a greater amount of information and knowledge, and then that changes how we approach different concepts. I'll get off my wild left turn. All right. So let's get back here. So now that we know it's DNA, now that we know that I can manipulate this crap, I can do really whatever I want, right? As long as I really don't have any morals, I could do almost whatever I want. There are a number of different laws that prevent a lot of stuff, but this is how we are able to alter things. And altering things is not a bad thing. We do it all the time with crops. We do it all the time with making vaccines, where you genetically modify organisms, those GMOs, right? And we can get greater yield in a crop. We can get a much more effective vaccine. Okay? There are a number of products created by our ability to genetically modify the DNA, inject it into something, a bacteria, put these bacteria in a vat, big vat, and then they can produce whatever the heck I want. And I can harvest that. And that's what they do with vaccines. And that's what they do with some supplements too, as well. Okay. I think there's a insulin that's made in this fashion. Okay. So there's a lot of commercial applications. We use green fluorescent protein. It's in jellyfish. And it's what codes their ability to be to be fluorescent in the dark of the water down deep. And we've isolated that gene, and I can take that gene and I can put it in the genome of a number of different organisms, and I can get things to glow. I can get the whole damn organism to shine in the dark if I want. For a lot of times, they tag a specific gene sequence with it. So when that specific gene gets expressed, it will also glow. And I haven't been able to figure out how to do a nice recording yet. I'm still going to try to figure it out for that What Darwin Didn't Know video that's in the syllabus. But that's what they did with that. They used this gene in jellyfish, and they injected it into the genome at various different spots in multiple other organisms. And then whatever that gene coded for, it actually would glow on the creature that they injected it in. And they were able to figure out what these genes make just by adding this little bitty protein marker on it. So GMOs can be fantastic, but I mean, for me, I think all the good, but then I think about what all the bad you can do with it, right? Because there's a number of bad things you could do with this, or potential bad things. What does DNA have? I think I have some pictures on a couple slides from now. I'm not going to ask you. I don't believe I had any pictures on the exam. Okay. I'm in the process of trying to put it together. But DNA contains 4 nucleotides, 2 purines called adenine and guanine. We represent with an A and a G. And 2 pyrimidine called thymine and cysteine. Okay. Adenine will always pair up with thymine. And cysteine always pairs up with guanine. Okay. And that's what they look like for grins and giggles. I'm not going to ask you. Okay. But that's what they look like. The great thing about DNA and this genetic code of A's and T's and G's and C's, every species has these. That's fantastic. I mean, this shows commonality. This shows common descent from something. And they're all in equal amounts. Okay, so A is always going to equal T, G is always going to equal C, so there's always an equal amount of all these, and with each species, it is unique. And here's on this thing here, I mean, this gives you the kind of the breakdown here, the semantics between a 23 and 23.3, they're all relatively similar, the same, okay? The dude that did this is Shargoff, okay? The amount of all of these things are the same for every member of a species, okay? Every dolphin has the same, every human has the same, every crab has the same, every species has the same amount of A's, T's, G's, and C's, okay? And it varies from species to species, all right? Okay. I'm not going to ask you, but there's 140 million base pairs, so the number of possible nucleotide sequences is 4 to the 140 millionth power. So that's a lot. Okay. A lot of variation. A lot of variation. That's fantastic. Okay. A lot of variation. A lot of codes. A lot of base pairs. But you know what the funny thing is? The majority of them don't code for anything. They don't code for a damn thing. And we don't know why. Well, we think we do. They think they may be these potential switches where they don't code for anything now, but at a potential time during our development, maybe it did code for something. So it's kind of fascinating, the vast amount of possible nucleotide sequences we have, and the vast majority of them don't code for jack squat. It's pretty interesting. So who saw all this nonsense? A doctor called Rosalind Franklin. She was one of the first. Her kind of claim to fame was she was able to take what's called an X-ray diffraction, and she, I think, perfected this process of getting an image of the DNA using X-rays, and it looks, it's pretty complex, and you're able to kind of see, you can kind of see the X there, and you can see, some bases there that are stained, and you can see it's in an X. So she was kind of the first to do this, pioneer this at a very successful rate. And so she was able to find these DNA fibers. She was able to get a picture of it, under these right conditions. So, she was able to kind of show that DNA was in this helical structure and that some portions of the helix are repeated. And you can see that from the picture. Similar base staining, similar base staining, similar base stainings. So that suggests that there is some repeated portions here. With Rosalind Franklin, she had a couple of other people that were working with her. Franklin, a colleague that she was working with, showed her work to Watson and Crick. The dude's name was Wilkins. I'm not going to ask you that. But he took her information and shared it with Watson and Crick because they were at the time working on what the heck does DNA look like, as was Rosalind Franklin. So they were able to kind of take her studies, parlay that into their own, and then they were able to come up with this double helical structure, complementary base pairs in between, G's and C's, T's and A's, hydrogen bonds holding it all together. So it was them, and I believe this was like in the 50s, and then 53. There you go. I don't know if I'd asked the date for that, but and then in the 60s they won the Nobel Prize in Science. Okay. They were able to come up with this double helical structure. Okay. Kind of a twisted format. Okay. Kind of a twisted format. Sugar phosphate backbone. Okay. sugar phosphate backbone. Okay, deoxyribose. Oops. Okay. Sugar phosphate backbone, deoxyribose. Hydrogen bonds held them all together. Okay. The DNA strands are anti-parallel. What does that mean? I called them a 5' end and a 3' end, and that just had to do with some chemistry that was at the ends of the strands of DNA. And this is what anti-parallel means. Okay, it's actually read in the 5' to 3' direction. So they were actually able to kind of see this makeup, how it was repeated, and actually it was repeated in an anti-parallel fashion. Okay. A's always go with T's, G's always go with C's. Building upon Chargaff's, Rosalind Franklin, and all of the work that was done before them, right, to prove DNA did exist. DNA is the magic. Okay. So we're able to take all of these experiments, all of these conclusions, and wrap it up into what we know now as the DNA model. Double helix, sugar phosphate backbone, complementary base pairs held together by hydrogen bonds. The great thing about hydrogen bonds, guys, right? Very weak, right? So we can break these apart really easily. And that's going to be something that's fantastic for DNA. So I can unzip the model of DNA, go in there, break those bonds, copy it, zip it back up, form the hydrogen bonds back, and I have preserved my DNA. Pretty cool.
The structure of DNA aids in the speed and accuracy of replication. Double-stranded DNA is a polymer of 2 strands of nucleotides which are hydrogen bonded to each other to form a double helix. Nucleotides are molecules that consist of a deoxyribose sugar, a phosphate, and one of 4 nitrogenous bases. The phosphodiester backbones consist of alternating sugar and phosphate groups. The nitrogenous bases include cytosine, thymine, adenine, and guanine. Cytosine forms 3 hydrogen bonds with guanine, and thymine forms 2 hydrogen bonds with adenine. This is referred to as complementary base pairing. The double helix will have one strand oriented in a 5' to 3' direction relative to the hydroxyl group of the deoxyribose sugar, and the other strand oriented in a 3' to 5' direction. This shows the anti-parallel nature of the DNA strands.
Okay. So now we've, oh my God, right? It's been a slow